Hey everyone, welcome back. In our last video, we used SAM tools to compress our alignment data into a sorted and indexed BAM file. We did a lot of heavy lifting in the terminal, but staring at text files is not how we actually find biological answers. So today we are going to do something incredible. We are going to visually look at the DNA sequences we just aligned. We are going to hunt for a mutation with our own eyes using an industry standard desktop application called IGV, the Integrative Genomics Viewer built by the Broad Institute. Okay, so let's step out of the terminal. We will open up IGV as you can see here and see what our data actually looks like. Okay, so unlike the tools we have used recently, IGV has a full graphical interface as you can see here. We don't run it in the terminal. You just go to the Broad Institute's website, download the desktop app for Windows, Mac, or Linux, or whatever system you have, and double click on the icon to open it. Basically, okay, it's basically a software. So when it opens, you will see a mostly blank screen. But here already a genome is loaded. But so normally you might see a blank gray screen. So don't worry, we just need to feed our puzzle pieces. Okay. So remember our jigsaw puzzle metaphor from previous videos. Before we look at our pieces, we have to show IGV the picture on the puzzle box. That is our reference genome. So you can load up your own reference genome. You can find that reference.fasta file that we used previously or your own itself. And IGV will instantly create a chromosome track at the top of the screen. Okay, so if you were working with human data, you could actually just select human HG38 from their built-in drop-down menu. As you can see, it's already loaded in here, so we will be just using that. Okay, it will automatically download the entire genome for your analysis automatically. Okay, so now let's load our data. Okay, so go to File and click Load from File, and you can find your file over here. So I'll take a sample.bam file. Okay, uh, and I will load it. Okay, and that file has been loaded. Here is a crucial pro tip basically. If you get an error in this step right now, it is because you forgot to run SAM2's indexing. Okay, in the last video, as I taught you. So IGV refuses to open a BAM file unless that tiny .bai index file is sitting in the exact same folder. Okay, so it needs the index to navigate the massive file quickly. Once it loads, you might still see a blank screen like this. Okay, that is because we are zoomed out completely and looking at an entire chromosome. So we have to zoom in. Okay, so at the top there is a search bar. You can type in any specific gene name. So I'll basically type ROC1. Okay, and or you can just click and drag on the ruler to zoom in. Okay, and keep zooming in until the gray screen transforms into a stack of gray rectangles. Okay, let's see what we can do here. Keep zooming in, keep zooming in, keep zooming in. And here you can see a stack of gray rectangles. Okay, these are your sequencing reads. Okay, so let's read this graph. The letters at the very bottom are your reference genome. Okay, each gray bar stacked above it is one of your 150 base Illumina reads. The bar graph at the very top is your coverage. Okay, it tells you how many reads stacked up on top of each other at the exact spot. If the coverage is 50x, it means the machine sequenced that exact letter 50 different times. Okay, now look closely at the gray bars. Okay, do you see a vertical stripe of color slicing through them? Maybe a stack of bright red T's or a green A's, as you can see here. Okay, if the gray bar is solid gray, it means it perfectly matches the reference genome. But if IGV draws a colored letter, it means the sequencing machine read something different. Okay, so if you see one random colored letter floating in a sea of gray, that is probably just a sequencing error. But if you see a solid column of color going all the way down through 50 different reads, then congratulations. Basically, you have found a single nucleotide polymorphism or SNP for short. You have discovered a real biological mutation. I do not see anything like that in here, but there are some gaps, but if you come across it, that is basically a single nucleotide polymorphism. And that is how computational biologists visually verify their findings. IGV is an incredibly powerful tool. You can use it to find missing chunks of DNA, which are called deletions, extra pieces of DNA, which are called insertions, or single letter changes. But here is the catch. We just looked at one tiny spot. A human genome has 3 billion spots. You cannot manually scroll through IGV to find every mutation in a cancer cell. That would take a lifetime. So we need to teach the computer to find these colored columns automatically, and we will be doing exactly that in our next video. Okay. And if you successfully loaded your first BAM file into IGV today, hit the like button and make sure you are subscribed. And let me know in the comments what kind of organisms you are studying right now. I'll see you in the next video.